right, so we now start the conversation about the adaptive immunity. Okay. So what is the difference? Why do we call the second branch of immunity adaptive?
It grows with us, and in a way, it's basically what we're exposed to, it adapts to, and we develop antibodies.
Yes. So this part of immune system is adapting to the microbes that we are exposed to. Okay, so it requires exposure, right? That's an important point. You can see however this thing works. It requires exposure, right? It doesn't develop response like innate immunity. It doesn't develop response without, it's not present, technically it is present, but it requires exposure to mount a proper response, right? But the feature of the adaptive immune system is that the responses of this system are highly specific and they targeted against a pathogen. So to some extent you can kind of compare them, make an analogy with broad and narrow spectrum antibiotics. Okay? So your innate immunity will respond to everything, but rather non-specifically. Your adaptive immunity will recognize a very particular microbe, all right? And of course that exposure requirement makes it slower. Obviously, it takes time for the cells of adaptive immune system to recognize the pathogen and mount the response, but generally the response is more effective because the cells, T and B cells, after they are exposed to the pathogen, they migrate into the circulation and they go with the blood, they go to every site of the body. So they can address the pathogenic attack, pathogenic injury everywhere. Okay, so it's systemic, slower and it's systemic. And the last but not least feature of that system is its ability to form immunological memory by a formation of a pool of so-called memory cells. Essentially, adaptive immunity remembers what you were exposed to. And if you encounter the same pathogen again, it will mount. So if you have the second exposure, it will mount the response faster, and this response would be more profound. Does that make sense? The adaptive immunity requires exposure. Responses are slower, but generally more effective, and they are highly targeted and highly specific. Also, adaptive immunity can form immunological memory. Which cells represent adaptive immunity? Those are lymphocytes. T and B lymphocytes. And each type of lymphocytes in turn represents a particular branch of adaptive immune response. B cells represent the humoral branch. Humor, except for being funny. What else does this word mean? Humor, related to human body. It's not used now, but in the old days, people had 4 humors. I remember bile, blood, phlegm, and third I don't remember. Humor. It's fluid. That's it. It's fluid. When we say humoral, refer to something in the fluid. In this case, humoral means that this branch is characterized by the production of antibodies, proteins that can be found in the human plasma. Okay? So the cells, just a quick overview, they produced and mature in the bone marrow, and when they are exposed, when they are matured, they go into the lymphoid tissue, lymph nodes mostly. B cells can be found in the germinal centers of lymph nodes. And when they are exposed to the pathogen, remember we talked about inflammation and how plasma seeps into the tissues and creates the continuous flow, washes away pathogens and washes them into the lymphatic system. So that flow of pathogens through the lymphatic system exposes them to B cells. And B cells, when they're exposed, they differentiate. They become plasma cells. And these plasma cells produce antibodies. Okay? We're going to review that process in more details. It's an overview. Does that make sense? Now, T cells, they also produced in the bone marrow, but they mature in thymus. That's why they're called T cells. And migrate in lymph nodes mostly and other lymphoid tissues such as Peyer's patches after maturation. And when they are exposed to the pathogen, depending on the type, there are different types, subtypes of T cells, they become different populations of T cells. Okay? They can be helpers, killers, pro-inflammatory cells, Th17, and regulatory cells. Now, some names say it all. Helper cells help other immune cells to become more effective. Killer cells directly destroy infected cells by apoptosis. Th17 cells increase inflammatory responses, and T regulatory cells play a critical role in stopping the immune response, in curbing it, because you want that war, for a limited time only. That makes sense. We're going to review all those features later. So T cells, T lymphocytes, represent the sub-branch that is called cellular immunity. Okay. So this is the general overview of adaptive immune cells maturation. They start in the bone marrow, produced in the bone marrow, but when they come out of bone marrow, they are naive. That's the official term. Naive cells migrate to thymus or stay in the bone marrow. The ones that are in thymus are T cells, the ones that are in the bone marrow are B cells. And once they mature, they migrate to lymph node. They are exposed to the antigen. I want to highlight it. They stay in the lymphoid tissue. Okay? And when they mature in the lymphoid tissue, after they are exposed to the antigen in the lymphoid tissue, after the exposure, only then they migrate into the bloodstream. Does that make sense? So they being exposed to the antigen in the lymph node, and after the exposure they get activated, go into the bloodstream. So understand this process. Be able to answer questions. Where is maturation? Where is exposure? Where are they produced? In which parts? Anatomical parts. But before we will start talking about individual branches of the immune system, we need to figure out what these cells respond to. And another key concept in adaptive immunity is the concept of antigen. Antigens are large, complex molecules that can initiate the adaptive responses. What can be large and complex molecules? Things like protein. Large? Sure. Complex? You betcha. DNA. Complex polysaccharides. complex polysaccharides with a complex structure, branching structure, okay? Does that make sense? Now, usually each antigen has many antigenic determinants, okay? So those are parts of the molecule that the immune system actually recognizes. Now, how I can look at us, all of us in this room. We all have a head. We all have 2 arms, 2 legs, right? We all look fairly similar. But I will recognize, I know that you're different, I can recognize you. Okay? How? You have subtle features, facial features, right? Height, hair color, that allow me to recognize you. That's how you recognize people, by the subtle features. You know? That's why sometimes, you know, you look at the description of the criminal, and they say, 6 foot tall male in the gray hoodie. Really? That's it? It's like half Cleveland. Another half 6 foot tall female. So it's kind of non-descriptive. But if you give more, you know, when you say to, I don't know, your friend, meet me, I'm going to be wearing, you don't say I'm going to be wearing clothes. That's what usually people do outside, you know. They don't go naked. You can say, you know, I'm going to be in a white t-shirt or, you know, pink pants, something like that. So give certain features. Same goes for the molecules. Immune system does not recognize the entire molecule. It recognizes features that are called antigenic determinants. Okay? And immune system develops unique response to each antigenic determinant. Even if the determinants are on the same molecule, look at this illustration here. The illustration of a determinant on the right, you can see the schematic representation of the different determinants with different shape. So each determinant will lead to the production of unique set of, say, antibodies. Does that make sense? Now, what is antigen? Is collagen that's in my connective tissues an antigen for my immune system? For my immune system. Is that an antigen for my immune system? Will my immune system build the response to my collagen? Unless I have an autoimmune disease, no. So you have to also appreciate that antigens have to be foreign enough to stimulate the immune system. Does that make sense? It should be foreign molecule. Does that make sense? Right? So in bacteria, in fact, bacteria have proteins on the surface. They're antigens. They foreign to our immune system. Sometimes, if you're really unlucky, certain proteins that otherwise are normally tolerated, like, say, milk casein, can cause allergy in people, and people will become allergic to milk. I'm not talking about lactose intolerance. That's a different story. I'm talking about allergy to milk. Okay, allergy to a certain protein that otherwise is not recognized by immune system. Does that make sense? So that's important. And again, antigens must be large and complex. Such large and complex molecules called complete antigens. Now, polyethylene is a molecule that is composed of many fragments of ethyl group. Okay, CH2 that is repeated millions and millions of times. That's what the grocery sacks are made of. Okay, so these molecules are large, extremely large. But look at this formula that I put over here in the top of the slide. Are they complex? Just repeated CH2 fragments. It's not complex. So polyethylene isn't an antigen. Although it's large, it's not complex enough. Does that make sense? Now, what about small molecules that are quite complex, but not large enough? Things like, say, amino acid or small hormone. Can they be antigens? If they're small, they're not going to be antigens. Such small molecules, sometimes called haptene. It's an incomplete antigen. Now, can this small molecule be enlarged? Yes. What you need to do is to add a carrier molecule to it. So take a haptene and you bind it to a carrier. Usually it's some protein, okay? And then now, it's like size difference. One thing is when, I'm walking, I'm rather regular size. Another story if I'm sitting on the elephant. I feel powerful in this case because I'm much bigger. Although it's just an elephant under my butt, but I'm much bigger. Overall, elephant and I represent a huge animal, right? Same is happening here. Haptene, and it is attached to the carrier, can stimulate immune response because together these two things are large enough. Does that make sense to you? And that is the way to make antibodies, industrially make antibodies to things like small hormones, like thyroid hormone, which would otherwise be unrecognizable. Does that make sense? Okay. Now we're going to talk about humoral immunity. So B cells, let's review what happens to them. So they are produced in the bone marrow as any other lymphoid cells. They are produced in the bone marrow. And they stay there for the maturation. Okay? So when they mature, cells produce different receptors, so-called B cell receptors, on their surface. Does that make sense? These B cell receptors, also called immunoglobulin D, each receptor can recognize a unique antigen. And we will talk how you can recognize practically everything in the world. So each receptor can recognize a unique antigen. Hypothetically, any of your proteins, your body proteins, can it be classified as an antigen? Does it have all the features of an antigen? Yes, it's large, it's complex. Does that make sense? So can you hypothetically, can you imagine that some B cell in the bone marrow somehow recognizes one of your body proteins? Technically, it's possible. It can have a receptor that would recognize certain body proteins. And if you will release such B cell into the body, what's going to happen? How do we call these problems? Nope. It's B cell that recognizes your own tissue, autoimmune response. Do we want autoimmune response? We do not. Cells that cannot differentiate self from foreign, you can see the examples. Here I'm going to circle them blue. Sorry, not this one. Cells that cannot differentiate self from foreign get eliminated. Okay? Such recognition in the bone marrow triggers apoptosis. these B cells die. Does it make sense? This is called a negative selection. Okay, negative selection of B cells leads to apoptosis of self-reactive cells, okay, or autoreactive B cells. Does that make sense? Okay, this idea kind of resonates with you, I hope. Now, obviously, you don't have just one B cell with a unique receptor, you have several of them. These cells that have the same B cell receptor, are called clonal population. Okay, sometimes they're just called B cell clone, not necessarily population. Maturation is over. Okay, maturation is over. You have millions and millions and millions of different clones of B cells. And each clone or clonal population has a very specific, very particular receptor on its surface. Does that make sense? Do you understand what I'm trying to say? So once they matured in the bone marrow, where do these B cells go? The lymphatic system. And they sit in the lymphatic system, right? And they wait. What are they waiting for? Antigen. So when they get exposed to the antigen, imagine there is an antigen that is recognized by this clonal population of B cells. This clonal population of B cells, that recognition, by the way, is called clonal selection. That population of B cells starts to proliferate and differentiate. It should be proliferate and differentiates. So what is proliferation? Become more abundant, yes, becoming more numerous. What is differentiation? Yeah, yes, to become more specific. So those that clone or clonal population of selected B cells becomes more numerous, and it also differentiates into so-called plasma cells. And this process, proliferation and differentiation of B cells, is called clonal expansion. Does that make sense? So once you have plasma cells start to produce antibodies. Okay. This is how antibody looks like. I'm just figuring out where would be the best time to talk about. So let's talk about antibodies now. Antibody usually is depicted having this Y shape on the left. You see? It's like a letter Y. So each antibody consists of 4 chains. 2 heavy chains, which one of the heavy chains I'm going to highlight with blue, and 2 light chains. One of the light chains I'm going to highlight with green. Okay, you can see that there are total 4 of them, 2 heavy and 2 light. Okay? The previous class taught me how you call the end of the fork. Thongs. Yeah. Like, there's a fork. You stick it in your steak. Whatever you stick. How do you call this? Huh? Prongs. Thank you. So the prongs of the fork harbor antigen binding site. Does that make sense? Does it make sense? Often these parts of the antibody, the parts that carry the antigen binding site, these parts, are referred to FAB, or antigen binding fragment. The stem region of the antibody is referred to as FC, or constant fragment or constant region. Does it make sense? Now, antigen binding site, here, FAB site, has to be variable. Why? Exactly, there are different antigens. So FAB has to be variable, has to change. Yes. Constant. Yes. Okay. So it has to be variable. You agree with me? And constant, well, you can see the name, it is constant. Turns out many cells of the immune system, they recognize and act depending on what constant region is here. So there are just, I think, 5 different types of constant region. Does that make sense, the idea? So you have part that binds to your immune cells, which is more or less constant, and part, FAB part, that binds, FAB part, that binds to the antigen. Let me put it here. So I'm going to write down, that's binding antigen. That must be variable. Right? Now let's assume that antibody takes about, how much should I say? Well, let's say 1,000 nucleotides. Okay? Now, wait, before we move to nucleotides, antibodies are what chemically? But proteins, yes. So proteins are products of what cellular process? Proteins are made in the process of the central dogma. Translation, yes, translation. And what is the template for translation? mRNA. And mRNA is synthesized in the process of transcription. And which, how do we call the tiny part of DNA that is transcribed in eukaryotes? Gene, right? Each protein, generally one protein, one gene, okay? So now we have an issue. Because each antibody has to recognize specific antigen. How many antigens are there? A lot. Look, we know about 1,500 species of bacteria in the human gut. Each of these species have, I don't know, hundreds if not thousands of proteins. So we talk about millions, well, probably billions or trillions of different antigens, right? Okay, let's limit at billion, just at billion. So that means that we have to produce billion different types of antibodies. If each antibody is encoded by its separate gene, then we're going to have a billion genes. Well, first of all, humans have 20,000 genes. Second, a billion genes would occupy 100 billion base pairs. Humans have 3 billion base pairs. And I just limited us to 1 billion, you know. What if we get +1? We bound. We don't have any antibodies left, right? How do we get around this? How do we produce such an enormous number of variants of antibodies? Yeah, but how can we make those classes? I don't know if you can see that. Those are 3 keys. They look quite similar, don't they? Really, I mean, it's like standard keys. One of them opens my office and 2 others are useless. Okay, because now we have like electronic keys, but nevertheless, at some point they were useful. So 3 keys. One opens my office. 2 others, I can stick them in the lock, but I can't open. And if you would look at them, they all have that, the groove, the little, the column teeth, all the kinds of stuff. And for me, I'm not, God forbid, I'm not a specialist. I was born with 2 left hands. They look very similar, very similar. But if you start to look closer, you see the differences. So they're like 2 teeth or 4 teeth, or, you know, groove goes here or there. So what do? We combine, right? We combine things. And we can have, I don't know, multitude of different keys. And our Bcells and T-cells, they do the same. This is called Vdj recombination. So before moving on, so receptors, okay, the B-cell receptors consist of 2 heavy, okay, heavy, and 2 light chains. Okay, light chains on this picture in the right bottom, they are white. Okay, in each chain is a constant region, which in this example, which is just an example, it's highlighted blue, so heavy parts are blue, and sorry, the constant parts are blue, and the variable parts have red, yellow, and green colors. Are you following me so far? These variable parts recognize antigen. Follow? Good? Blink twice if you don't. Okay. Now, each heavy chain has 44 variable, 27D, 6J, and 2 C gene fragments. Okay, let me stop for a second. This is the part of the chromosome that represents genes for the heavy chain. This is the part of the gene. This is the part of the gene. This and this. Do you follow me so far? During transcription, one part, one V segment, is co-joined by one D and one J segment. Does it make sense? Now you have 44 different V's, 27 different D's, and 6 different J's. The total number of variants, if you combine those V, D, and J fragments, is 3 multiplied by 10 in the power of 11, which gives us 300 billion. It's enough. Does that make sense? I usually use analogy of clothing. Okay? Now, please don't laugh about color selections, but imagine that I have, well, okay, I have 3 different t-shirts. I have 2 different shorts. I have 2 different shoes. It's not a lot of clothing, but combining them, I can have different outlook. Does that make sense? That makes sense. That's what happens here. A combination between those genes, Vdj recombination, provides you with enormous number of variable regions, FABs, on the antibody. Does that make sense? Okay, so that's how we produce so many variants. Now, types of antibodies. We're kind of moving on towards the functions of plasma cells. So remember, plasma cells are the cells that produce antibodies, not the B cells. B cells have to differentiate in plasma first, okay? So B cells differentiate, become plasma cells, and plasma cells will produce antibodies. There are 5 major classes of antibodies. I'm going to start with the boring one. It's IgD, immunoglobulin D, which is the B cell receptor. That's it. Okay. It's kind of written here. B cell receptor. Next one that I wanted to mention is IgE. G has quite a unique feature. Okay, it's allergic antibody. So it is produced during exposure to the allergen, and it sticks. So the constant region, this part of IgE, sticks to the cells like basophils and mast cells. And when it interacts with antigen, it causes degranulation. We will talk about allergy response mechanism. Don't worry about it. What you have to appreciate is that IgE is the antibody responsible for the allergy development. Okay, good? IgA. This antibody forms a dimer. It always exists in the form of a dimer. You can see 2 antibody molecules co-joined at the constant region. Co-joined here. So this IgA is a secretory antibody. Okay. So it is found in the secretions, mucus secretions. We're going to find mucus secretions in the human body. Yeah, the entire respiratory tract, okay? Mouth, throat, trachea, bronchi, where else? Not mucus, it's a serous secretion, no. Other mucous membranes. Which are? So respiratory, Which epithelium? Not all epithelium is exposed to outside world. So respiratory, we figured out. Which another system is pretty much a gaping hole in the human body. Yeah, digestive, it's a hole, right? Starts at the mouth, ends at the anus. Mucosal surface all over the place. So IgA is indigestive. Another. Exposed to outside world. I mean, we can get like probably in prison for several days if we actually expose it to outside world. Reproductive. It is exposed to outside world. There's a mucous membrane. There are IgAs there. So they sort of a first line of defense. They protect against invasion by epithelial layer. Does that make sense? Okay. IgM, gigantic immunoglobulins, okay? These are pentamers, so you can see 5 molecules that are co-joined. And first of all, this is the first antibody that is produced in response to infection. IgMs are always produced first, okay? They can activate complement, okay? They can precipitate. We'll talk about mechanisms, but they can activate complement. And finally, the most numerous antibodies, IgG, right? Immunoglobulin G. So this antibody, also it's monomer, It is a second antibody that's produced in response to infection after IgM. It activates complement, and what is important? It is it can cross placenta. Okay, so it provides that passive immunity to the fetus. Not only to the fetus, to the newborn. It gives some immunity to the newborn. So newborn is born with mother's antibodies. And mother continues to supply antibodies to a newborn via the milk. However, in the milk, you're going to find IgAs. Okay? That makes sense? I believe that sweat is not really correct about IgAs. And think we produce IgAs in the sweat. Okay. So, primary response. What happens? How humoral immunity responds during the primary exposure. You have B cells, okay, with IgD or B cell receptor on the surface. This particular type recognizes the antigen, and it starts to proliferate, clonal expansion, and differentiate. B cells will differentiate into the plasma cells. Plasma cells will produce antibodies. And B cells will also produce a population of memory B cells. Okay? Special clone that remembers that exposure to the pathogen. Okay? Circulating memory B cells are already primed. They're ready to quickly respond to the pathogen again. All different kinds of antibodies are going to be produced. Well, mainly IgM, IgG, and IgA will be produced specifically on the mucous membranes. Secondary response. When memory B cell is exposed to the pathogen again, it will proliferate and differentiate, producing a number of plasma cells and memory B cells again. You may ask, wait, what's the difference between primary and secondary response then? It is timing. Okay, look at here. So you have at point 0, it's a point of primary exposure, and you can observe increase in antibody production that occurs about a week after the exposure. Does that make sense? And then number of antibodies goes down. When the person is exposed to the pathogen again, antibody titers elevate tremendously. And moreover, they not only elevate, they stay elevated for a longer time, giving you protection. Does that make sense? However, if you expose the person to a different pathogen, the antibodies levels will be still low. So the more exposures you have, the higher antibody titers you're gonna expect. Does that make sense? Tell me if I'm going too fast. I really want to talk about something, but probably not today. But tell me if I'm going too fast. Really, slow me down. Humoral immunity can be of 4 different types. It can be divided based on I don't know. I have no words how to formulate. Okay, let's start from a different point. Active versus passive immunity. So active immunity, when your immune system is directly exposed to the antigen. Does that make sense? So you receive the antigen. Passive is when you simply receive antibodies. It's called passive transfer. Okay, that's passive immunity. Natural immunity is when everything happens the nature's way. I hate this expression. Nature's way. Artificial is when humans involved. Does that make sense? Now, when and how can you be exposed to the antigen? What can result in the exposure? Vaccine. Okay, bingo. Vaccine, is that inactive? Is that active immunity? So you're exposed to the antigen. Yes, it is. Is that natural or artificial? Artificial. Perfect. So it's active artificial vaccine. How else? Yeah, you can be infected. Is that active? Surely, but it's natural. Okay? Does that make sense? So, infection is active, natural immunity. Okay? Vaccine, as we discussed, active, artificial. Now, what is passive natural? If you can look at without looking at. Yes, you can consider transplacental, yes. Milk, yes. Mother to a child. That's pretty much the only example. I'm not aware of any natural way of blood transfusion. It always involves humans. And that is transfusion of not necessarily blood, well, sometimes it's whole blood. People with severe immune deficiencies, they often have to receive the whole blood, okay? But in some cases, you receive antibodies. So passive artificial will be tetanus shot. Or if, God forbid, you get bitten by a snake, what do you get? What it is? What is that? Nope. Nope. Venom is a protein. Well, venom is not a protein. Venom is a fluid. But the acting compound is a protein. So this protein, this venom, is injected into horses. Horses, big animal. So you inject venom, they don't die, but they develop antibody response to it. Then you can, horse is a big animal, it has a lot of blood. So you can bleed the horse repetitively, isolating antibodies, and then using these antibodies to inject into humans when they get exposed to the venom. Does that make sense? Speaking of which, if you've read like stories about medieval stuff and some kings and queens and aristocracy, they sort of built up tolerance to the poisons. You may have heard about it by taking the larger dose of a poison with a time. What they essentially did, they exposed the immune system to a poison. Does that make sense? And they accumulated antibodies, so they trained the immune system. So eventually, somebody, poured that poison into their drink, they drink it, the immune system shuts it down completely. Not every poison works like that. Don't try it at home. I mean, ethanol is the poison, it doesn't work. You can expose yourself to elevated amounts of ethanol, you're going to die. Well, you're going to become alcoholic first. So how antibodies, it works with the proteins, How antibodies work. What they do, I mean. How they can protect. First of all, antibodies do not kill anything per se. Never. They never kill directly. What antibodies do is they bind to the pathogen and often tag it. Do you see what I'm trying to say here? For instance, they can bind to the surface of the microbe and activate complement. Okay? Or They can bind to the surface of the microbe and increase phagocytic responses. Or, remember opsonization? So now we know two things that can opsonize. Complement can opsonize, and antibodies can opsonize, increasing attractiveness for phagocytes. They kind of screaming for the, like, you know, like these things in the department stores on clothing and shoes. They have this plastic things. If you try to remove it yourself, there's going to be, I was told, never tried it, ink sprayed all over the place. This is pretty much like antibodies, okay? Antibodies label the pathogen. They tell, this is bad, kill it. They can increase inflammation, of course, okay? Especially in allergy responses. Now, of course, things like membrane attack complex will lead to cell lysis, but it's not antibodies that do it, it's complement that does it. Does it make sense, right? What else can they do? They can bind to things like virus and prevent it from doing anything. Okay? So they don't kill the virus, they don't disassemble the virus. But when virus is surrounded by antibodies, it cannot infect because recognition of a receptor is totally blocked. Right? That makes sense? So neutralization. They can agglutinate. For instance, this IgM shown in this picture, right? It's going to bind multiple cells and clump them all together, limiting their motility or making them an easier target for macrophages. Okay? And they can precipitate. That relates to our discussion of the snake venom. Antibodies can bind multiple proteins and clump them all together, so they're gonna produce this, often they're called immune complexes. And you can imagine, the bigger the complex, the easier it is for a macrophage to destroy it. Does that make sense? So let's review mechanisms. Antibodies can neutralize. Antibodies can agglutinate. Antibodies can precipitate. They can fix complement, which leads to the cell lysis. Okay? Fixation of complement will also lead to increased inflammation. And antibodies per se generally can opsonize and stimulate phagocytosis. Does that make sense? You have to know these mechanisms. You have to be able, like if I ask you, can antibodies lyse a bacteria? Tell me, no, they cannot. Okay. If I ask you, what can opsonize the bacteria? You tell me it can be a complement or it can be an antibody. They both can opsonize, increasing. What is opsonization? Increasing the attractiveness of a pathogen to phagocytes. Does that make sense? Okay. Oh, Jesus H. Christ. Well, I'll say a few words. I'll let you go soon, but that is a tough one. Not that I'm discouraged. No. It's actually a great concept that explains why transplants don't work as we want to. Major histocompatibility complex. Histo. What does that mean? Tissue. So it's a complex of proteins, several protein molecules, that define compatibility between your immune system and your cells of the body. Essentially, it's this MHC. Let me refer to it as MHC. MHC molecules serve as labels that signal self or foreign. Okay? There are two types of MHC molecules, type 1 and type 2. Type 1. Now, before we move on to type 1, type 2, what MHC molecules do, they present antigens. Now, imagine that the antigens that they present are peptides. So MHC presents the peptide on the surface. And if everything's fine, one peptide is present. That's the marker here. If cell is somehow changed, if it's infected, or if it's cancerous, the antigen presentation changes. It's like a flag that goes from normal to infected. Does that make sense? Self to foreign. Okay? So we're going to start with that tomorrow, and